So I know that this is a bit early, but our next speaker will be Dr. Raleigh Linville. Dr. Linville is a soon-to-be incoming postdoc at the Broad Institute, and he actually just recently successfully defended his PhD as a graduate student of the Searson Lab here at Johns Hopkins. A dedicated scientist and researcher, Dr. Linville was a recipient of the 2018 National Science Foundation Graduate Research Fellowship Program for his focus on developing tissue engineering models of the human blood-brain barrier for treating central nervous system diseases. Even during his formative years, Dr. Linville was an inspiration for helping others as he led his local Boy Scout troop and community as an Eagle Scout to help develop solutions for pediatric patients that allow them to read and write from their hospital beds. Dr. Linville has also served as a graduate research mentor to multiple undergraduate students in the Searson Lab, and I've heard nothing but good things from them. It is my pleasure to introduce Dr. Raleigh Linville, and it actually looks like he's connecting to audio right now.
Hello, it's a pleasure to be here. Can you hear me?
Yep, you're all good.
1 sec, I'm still getting set up because we're running a few minutes early.
Oh, absolutely.
Okay, can you see my screen?
Yep, looks great.
Okay, great. It's a pleasure to be here today to talk more about my PhD work. Thank you for that introduction. So today I'll be talking about tissue engineered microvessel models of the human blood-brain barrier. This is research that I've conducted over the last nearly 5 years in the Searson group at Johns Hopkins University. So I first wanted to start with an introduction to the human blood-brain barrier. The human BBB is a specialized network of blood vessels that separates the bloodstream from the brain tissue. And like the human brain, the BBB is highly dense. We see a very dense network of capillaries within the human brain nearing a length of around 600 kilometers. And these capillaries maintain very close proximity to neurons. And proximity between capillaries and neurons allows for a very efficient exchange of nutrients, things like glucose and oxygen, and efficient removal of things, of waste products as well. And so as human brain activity is highly dynamic, the BBB is able to mediate a process known as neurovascular coupling, where nutrient delivery is matched to regions of local brain activity. And this can be visualized with imaging modalities like fMRI as shown in the bottom left. The BBB also acts as a layer of protection. It shields cells in the brain from toxins and pathogens that are present within the bloodstream. So historically, this was discovered by taking a dye and injecting it into the bloodstream of an animal and observing that the central nervous system, so the brain and the spinal cord, do not take up that dye. So we can see that in a guinea pig embryo on the left. At the same time, this barrier is also able to restrict many damaging things from entering and hurting your neurons. But at the same time, many therapeutic compounds to treat brain diseases are restricted from entering into the brain, which is a major barrier to the treatment of brain disease. At the same time, the human BBB is also obviously human specific. Recently, a lot of this has been unraveled at the gene and protein level in terms of differences in between humans and animals like the mouse model. And so importantly, during diseases, the BBB and the brain are linked. This is both structural and functional. So when you have brain health, you have blood-brain barrier health. When there's blood-brain barrier dysfunction, you have brain dysfunction. And these can propagate one another. And so in many brain diseases, there are both structural and functional changes in the BBB. And one example of this is neurodegenerative disease. So here we see in blue the accumulation of a toxic protein known as amyloid beta on blood vessels occurring during Alzheimer's disease. However, across many brain diseases, things like trauma, brain cancer, stroke, and autoimmune disorders, there are changes in the blood-brain barrier that occur. And collectively, these diseases are very common, as over 15 million Americans suffer from serious neurological disease. And so there's multiple tools at hand to study the blood-brain barrier during health and disease. Commonly, animal models are used as they're inexpensive and can allow very high-resolution studies. However, as discussed previously, there are interspecies differences that may alter the relevance of these findings. So to provide human specificity, post-mortem human tissue can be used. However, there's very limited access to this tissue, and it likely only partially represents what's actually going on in healthy brain tissue. So to study healthy brain tissue, different imaging modalities can be used, things like MRI to study the brain. This can be done in real time and also has very high field of view. So you can look at entire brain regions over short periods of time. But unfortunately, these technologies do not have very high resolution to study what's happening at the level of single cells or single microvessels. So an alternative approach is to use cell culture. So this is a so-called in vitro approach where we'll model the blood-brain barrier outside the human body. And the one major limitation to this approach is that when we grow cells outside the human body, they do not necessarily represent the structure and function of native tissue. And so with simple cell culture and all these other techniques combined, there are still many fundamental and clinical questions about the BBB that cannot be fully addressed. And so the goal of my research and the broad goal of the research in the Searson group is to develop tissue engineered models of the human BBB to overcome many of these limitations. So this process can be broken down into 3 steps. First, we have applying tissue engineering principles to build models of the BBB. Second, we have validating models by comparison to our current understanding of the human blood-brain barrier. And third, we have applying our models to answer various fundamental and clinical questions. And so I'll discuss several applications of these models throughout the presentation, including using models to develop new drug delivery strategies, as well as using models to understand and treat blood-brain barrier dysfunction that may occur during brain diseases. So the work in the Searson group obviously can broadly be mapped to these 3 stages, from engineering to validation to application. And we see in each of these circles some of the various things that we do in the lab to engineer, validate, and apply our models. For my talk today, I'm going to first start with an overview on the engineering and validating of tissue engineered BBB models. I'll then spend some time highlighting the distinct advantages of tissue engineered BBB models. And third, I will highlight applications of tissue engineered BBB models. Okay, so first, I'll talk about my work to engineer and validate blood-brain barrier models. And some of the important things I'll talk about are the choice of a cell source, the choice of a model type, as well as showing the validation that's been conducted. And so when we think of designing the blood-brain barrier in the lab, we're obviously inspired by the native structure and function of the human BBB. So I want to talk a little bit more about structure and function. If we zoom in to the structure of the BBB, we see a cross-section of a blood vessel. It's lined by brain microvascular endothelial cells, or BMECs. And surrounding these BMECs, we have a specialized extracellular matrix called the basement membrane. Additional cell types involved include pericytes and astrocyte end feet, which cover the surface of the basement membrane. And along the arteriovenous axis, so from arterioles to capillaries to venules, there are changes in not only vessel diameter, but also in the different support cells that surround these blood vessels. Additionally, the cells that line these blood vessels from arterioles to capillaries to venules are intrinsically different. And this recently has been discovered using single-cell RNA sequencing approaches, finding that some specific genes are only expressed in certain zones of this hierarchy. So the BBB has a lot of different functions. The one I'm going to mostly focus on today is the regulation of permeability, or how compounds are able to enter and exit into the brain. And so there are really three intrinsic properties of BMECs that enable them to regulate barrier. The first is the expression of tight junction proteins. These are proteins that are expressed between endothelial cells, and they reduce the paracellular permeability, or the passage of compounds between cells. Second, we have efflux pumps. These are expressed on the luminal surface of BMECs, and they basically pump compounds that passively diffuse into the BMEC back into the bloodstream. And this is a mechanism to reduce the transcellular permeability, or the passage of compounds through brain microvascular endothelial cells. And third, we have uptake transporters. These are an array of transporters which are located at the plasma membrane, and they shuttle important compounds like nutrients into the brain. And together, these properties dictate the permeability of compounds. For example, compounds that are very important to neuronal function, like amino acids and glucose, rely on uptake transporters, while other compounds that we know are able to enter the brain, like caffeine, are able to passively diffuse into the brain due to their low molecular weight and because they're not recognized by efflux pumps. On the other hand, there's compounds like the chemotherapeutic agent doxorubicin, which is not able to widely enter the brain, and this is because it's recognized by efflux pumps, which pump it back into the bloodstream. And then many large molecular weight compounds like antibodies or components of blood like serum albumin have very low permeability due to the expression of tight junctions. And so historically, in vitro models of the BBB are highly reductive in terms of replicating the structure and function of the BBB. For example, to screen the permeability of drugs, a combination of MDCKs and a transwell assay have been used. So MDCKs are epithelial cells derived from Cocker Spaniels, and these cells can be grown in a transwell assay, which where we see this porous membrane, and above and below this porous membrane, there's a media reservoir. And so you can add the drug on top of the cells, and you can measure the rate at which compounds enter into the basolateral chamber to determine the permeability of a drug. And this approach is obviously very high throughput, it's inexpensive, and for many compounds, this is a very accurate way to screen the ability of a drug to enter into the brain. But obviously, this approach does not fully recapitulate the structure and function of the native human blood-brain barrier. So my research has been to utilize advances both in cell source as well as in model type to build kind of a next generation of tissue engineered BBB models. And so in terms of the cell source, I will show that I use iPSC-derived brain microvascular endothelial cell-like cells, or iBMECs. And in the model type, I'll show my work using 3D microvessel models. So iBMECs are differentiated from iPSCs, or induced pluripotent stem cells, using published protocols. With these cells, we see expression of critical tight junction proteins like Claudin-5 and occludin. We see expression of uptake transporters like LUT1, which shuttles glucose into the brain. We see expression of efflux pumps like PGP, as well as the expression of endothelial markers shown at the bottom. And if we take these cells and we grow them in a transwell assay, we see they recapitulate critical barrier functions. For example, their trans-endothelial electrical resistance, or TEER, matches recordings in animal models, while other cell lines have very low recordings. And if we measure the permeability of compounds across the monolayer, we see robust paracellular permeability, where we get really low permeability values of compounds like dextran. And we also see efflux activity as evidenced by a directionally dependent permeability of efflux substrates like rhodamine. Additionally, with these cells, we can conduct bulk RNA sequencing to study changes in gene expression due to differentiation. And shown here is a volcano plot that summarizes changes in gene expression. Each dot represents a single gene. On the x-axis, we see the log 2-fold change in gene expression And on the y-axis, we see the significance of the result, where something at the really top right of this volcano plot is both a gene that's heavily enriched in IBMECs as well as statistically significant. And so across many replicates and 3 different cell lines, there's many shared genes that are upregulated by differentiation, including some of the proteins I just talked about, things like GLUT1, which is a uptake transporter for glucose, as well as CDH5, which is the gene encoding V-cadherin, which is an endothelial marker. And these genes that are upregulated in IBMECs are associated with a variety of biological pathways, things like ECM organization, adherence junctions organization, blood vessel development, VEGF signaling, among others. And so together this shows that IBMECs are able to recapitulate various aspects of brain microvascular endothelial cells. And so next, we use a 3D microvessel model to apply these cells. And so with this model, we take a 150 micron diameter rod and we suspend it within a PDMS-based microfluidic device. Around this rod, we gel type 1 collagen. After gelation, the rod can be removed and it leaves behind a bare channel that can be processed with cross-linkers and ECM proteins. Ultimately, we can add cells into this channel and these cells assemble into a 3D tubular structure. And so this is the microfluidic chip. And this is an example of a perfusion system to continually perfuse this tube with media. And here we can see a 3D reconstruction using confocal microscopy of this tubular structure. And here the cells are labeled green, enabling us to see them easily. So with this platform then, I've done a variety of experiments to validate the function of this model. One of the most common approaches is to measure permeability. To do this, we take some sort of fluorescent dye and we perfuse it through the lumen of the microvessel. And so shown here are 2 videos following the perfusion of the lumen with lucifer yellow. We see on the left a HUVEC vessel, which is an endothelial cell that is not brain specific. And we see that lucifer yellow readily exits this blood vessel. It's very leaky to lucifer yellow. While on the right, we see IBMEC microvessels that display very low permeability, nearly 20 times lower. This shows that this model has very low paracellular permeability. If we use confocal microscopy, we can see why these cells have lower permeability, and it's because they express things like tight junctions, Claudin 5 and occludin, shown on the left, and the models also express uptake transporters and efflux pumps, as shown on the right. The 3D models are also responsive to inflammatory cues. We can perfuse with tumor necrosis factor alpha, which is a cytokine, and see that models upregulate at the protein level surface adhesion molecules like VCAM and ICAM. And these molecules are upregulated to mediate the adhesion of immune cells to the endothelium. And indeed, we see that with TNF-alpha, immune cell adhesion is increased. We also see a degree of maturation that occurs within the model over 6 days by the deposition of basement membrane proteins. And we can also construct this model across a variety of different iPSC sources. In terms of other engineering complexities, I've worked to incorporate other cell types. So while most of my work is focused on BMECs, I have incorporated pericytes, which are another important cell type of the BBB, into this model. Compared to 2D assays where you have very indirect interactions between pericytes and endothelial cells, in our 3D models, we can get direct cell-cell interaction. I've also have ongoing work to study the influence of metastatic breast cancer cells on the function of IBMECs in this platform. Another angle is to engineer hierarchy. So the goal is to develop microvessels that have a variety of different diameters. And so this is very challenging to do in 2D. However, in 3D, we can pattern adjacent microvessels and using growth factors, promote the process of angiogenesis, or the formation of new blood vessels. And these new blood vessels that form between microvessels maintain barrier functions like low permeability, as well as expression of important proteins. Okay, so now I want to highlight some of the advantages of tissue-engineered BBB microvessels. First is that these microvessels allow us to recapitulate many environmental cues that are present in the native BBB. This is things like shear stress, direct cell ECM interactions, cylindrical geometry, and transmural pressure. And compared to other complex models like microfluidic chip models, in 3D microvessels, we can recapitulate a lot more of these cues. A microfluidic chip model like the paper that's cited here really is only able to achieve just application of shear stress. It doesn't have a lot of these other complex properties. And so because of all these differences in microenvironmental cues, we wanted to study how these differences affect the phenotype of models. And so we used bulk RNA sequencing to directly compare gene expression between 2D and 3D. And so we see that there's hundreds of differentially expressed genes between 2D and 3D models. There's around 400 differentially expressed genes that are enriched at least twofold by the 3D microenvironment. And compared to previous studies on shear stress, it's about twice as many genes. So this shows that 3D microenvironment induces a very dramatic effect on gene expression. There's also very limited overlap between genes that are upregulated by shear stress versus genes that are upregulated by 3D microenvironment. And so this suggests that there's a very distinct change that's induced by 3D microenvironment. And indeed, many of the genes that are upregulated are associated with biological pathways that are quite unique from ones that are upregulated by shear stress. This includes things like extracellular matrix organization, response to growth factor, regulation of angiogenesis, and regulation of coagulation processes. And so matching these changes in gene expression, I've also validated that there's changes in protein expression. 3 protein examples are shown here that are upregulated in 3D, and all 3 of these proteins are observed to be expressed in the mouse blood-brain barrier. And in terms of functions, we also see some differences. In 3D microvessels, we achieve much lower permeability values, and permeability values are stable over a 6-day period. While in transwells, you have higher permeability, and it increases over time, suggesting that these cells are really changing during the length of culture. If we then look at these permeability values that we achieve with the 3D microvessel system and compare them to published literature, we see very close proximity. So we achieve around 2 × 10⁻⁷ centimeters per second permeability of loose free yellow in our microvessels. And this matches recordings that have been done in rat brain peel venules. And compared to other approaches that use primary cell lines, we're able to achieve a much lower in physiological permeability values. Okay, so with these various advantages in mind, I want to conclude by highlighting some of the diverse applications of BBB microvessel models to answer biological and clinical questions. And so the first application I'll highlight is to understand how the BBB maintains barrier function during cell turnover. So previously, we've been able to track turnover by monitoring if cells go through processes like apoptosis or mitosis using phase contrast microscopy. Interestingly, we can easily see there's tons of cells in microvessels that go through these processes, but there's no change in paracellular barrier function when these processes occur. And what that means is that cells are able to divide and die without compromising local permeability. And so we were very interested to study how are cells able to divide and die while maintaining barrier properties. And this is a very challenging question to answer using traditional techniques. So we developed a model using an iPSC source that has GFP tagged tight junctions. And for the first time, this model allows us to track tight junctions in real time during all stages of cell turnover. And so here in the middle, we see traces of individual cells over 1 hour, a quiescent cell at the top, a mitotic cell in the middle, and an apoptotic cell at the bottom. And from these traces, we can measure all sorts of morphological metrics, things like area, aspect ratio, the number of cell neighbors, and the instantaneous speed of cells. And we can classify cells into various populations. And from this, obviously there's a wealth of data that we've accumulated. A lot of the data is summarized in this heat map, where the color represents both the direction of change of a metric as well as the statistical significance of that change. And not going to go too in depth into these, but I'll highlight a few points. The first being that quiescent cells are very stable. And this makes sense, right? There's some minute fluctuations in area and perimeter over time, but overall, the majority of cells in a microvessel have very, very stable morphology. We see that during the process of mitosis, cell division occurs without disrupting the area or perimeter of cells that are neighboring a mitotic cell. And during apoptosis, very interesting result, while the apoptotic cell obviously shrinks in size, the neighboring cells do not directly compensate this loss in area. They themselves also have a loss in area during this process, which suggests that the entire monolayer collectively displaces the lost area from an apoptotic cell. And so obviously this is just a few key insights, and there's a lot more details here that I'm not able to go into. Additionally, we can study how the BBB responds to physical and chemical injury. We can induce physical injury using a high-intensity laser and ablate a hole in the endothelium. This kind of models something like hemorrhagic stroke or traumatic brain injury. We can image how the endothelium repairs over time, and we see this process is mediated by cell migration, not by the proliferation of cells. We also can model things like chemical injury. Use the oxidative agent metadione. This causes cells to accumulate reactive oxygen species. And this is kind of a hallmark of many brain diseases, including things like neurodegenerative disease. And we see that this chemical induces a very unique change in the microvessels, where it promotes the detachment of the endothelium from the extracellular matrix. And this process happens without increases in apoptosis. So this is a very unique mechanism of oxidative injury that we're in the process of further exploring. Another separate application I want to touch on is studying the mechanisms of hyperosmotic BBB disruption. And so I've talked about that the blood-brain barrier regulates permeability into the brain, and obviously this is a major hindrance to the delivery of drugs. So one approach to increase drug delivery is to use compounds like mannitol. Mannitol is able to shrink endothelial cells when you inject it into arteries that supply the cells of the brain. And after this mannitol injection, you can then perfuse large therapeutic agents, things like antibodies and gene therapies, which are then able to more readily pass into the brain over a very short period of time before the barrier is able to repair itself. And so we're really interested in studying this process in a little more detail. There's a lot of unknowns about this process, and it's very hard to image in mice. And that's because in mice, from this video here, we see that opening occurs predominantly within the hippocampus, which is the region circled in red. And that means it's inaccessible from multiphoton microscopy approaches. So we really don't have good resolution to see what's happening at the level of single microvessels in the mouse brain during this process. So we basically studied this process in collaboration with a professor of radiology, Professor Piotr Walczak at University of Maryland. And so to model this process, we applied clinically relevant doses of mannitol into microvessels. At the top, we see a control microvessel. It doesn't display any leakiness of dye. However, with 2 and 5 minute mannitol doses, we see transient disruption and leakage of dye. It's a very dramatic effect that quickly reverses. And if we zoom in on the sites of disruption, we can actually visualize pathways of dye traveling between adjacent BMECs. And this is what we call a focal leak. It's this discrete location of paracellular increases in permeability, or the passage of dye between endothelial cells. And what's very interesting is, despite the fact that there's very dramatic increases in permeability induced by mannitol, it really only is caused by a very small fraction of all junctions being disrupted, which we predict to be around 0.04%. And so this is some view of how mannitol disrupts the BBB, and is important to understanding. It's very important to understand this to motivate improved techniques for drug delivery into the brain. The last example that I'll touch on is to develop a novel approach to increase drug delivery into the brain. And so this is work that we've done in collaboration with Professor Piotr Walczak and Professor Kalina Hrstova at Hopkins. And we've been very interested to use what are known as membrane active peptides. And so membrane active peptides are able to accumulate on membranes, and if at high enough concentration, they can actually form pores in membranes to kind of disrupt lipid membranes. And so the kind of standard membrane, the most well-known and well-studied membrane active peptide is melittin, which is the main protein component of honeybee venom. And so we wanted to study melittin, as well as many variants of melittin that have even higher membrane activity, to see if these compounds could in any way be used to transiently increase drug delivery into the brain. And so not only did we want to develop a new approach, but we also wanted to develop a new pipeline to screen approaches in the future that can be used for drug delivery. And so our pipeline consists of 3 steps. We first take our membrane active peptides and we conduct cytotoxicity screening. This allows us to determine a range of doses that we can then apply within tissue engineered models to further optimize dosing. We also, in the tissue engineered model, can basically better study mechanisms of how opening occurs. And lastly, then, based on the results in tissue engineered models, we can then apply this technique within mice models to assess neurological safety of the approach. And so to summarize some of the key findings, when we apply melittin and its variants within tissue engineered microvessels as bolus doses, we see transient disruption of the BBB. In green is the dye 500 kilodalton dextran. We see it very dramatically increases in permeability, and then that effect wears off quite quickly. We've also screened many melittin variants to better understand the structure-function relationship between a membrane active peptide and its ability to promote BBB opening. And additionally, using microvessels with fluorescently labeled tight junctions, we've studied kind of the mechanisms of opening and found that, very similar to mannitol, melittin is able to induce paracellular leakage by disrupting cell-cell junctions. And kind of lastly, in collaboration with Professor Piotr Walczak, we've applied melittin as intraarterial injections in mice, and we see that infusion results in the rapid leakage of the contrast agent gadolinium in the mouse brain. And this completely is reversed within 24 hours and is also not evident at 1 week, showing it's a very transient effect. We also then use immunohistology to study neuroinflammation and neuronal damage, and found no evidence of any sort of acute or chronic damage to the brain tissue using this approach. And so overall, this work shows that 3D microvessel models can be used to translate drug delivery approaches towards clinical applications. Okay, and so to summarize, this work kind of highlights the diverse applications of tissue engineered models, and also shows some of the unique advantages. With a tissue engineered model, we have unique gene and protein expression, we have enhanced barrier properties, we have very high spatiotemporal resolution, And together, this has kind of enabled our lab to study many of these very interesting fundamental and clinical questions. And with that, I'll conclude, and I'd like to acknowledge all the members of the Stearson Group for their contributions to many aspects of this work, as well as many excellent undergraduate mentees, many who are now pursuing their own PhDs or medical degrees, and then some that are obviously still working in the lab, as well as our lab's collaborators. With that, I can take any questions people have.
Thank you so much, Dr. Linville, again. Any questions from the audience about his work?
I have a question. So yeah, you talked about like learning about how to build a tissue engineered BBB model. So how are you gonna take this knowledge to your postdoc, and what are you gonna do there?
Great question. So for my postdoc work, I'm going to be studying kind of more about the details of zonation of the blood-brain barrier. So I briefly talked about how there's differences between arterioles, capillaries, and venules. And so in my postdoc work, I hope to kind of lay some of the groundwork to start building blood-brain barrier models that are specific to arterioles, capillaries, and venules to enable more detailed studies.
All right, if there are no more questions, thank you again, Dr. Linville. I think we all at least found the presentation very interesting, and we wish you good luck on your future.
Thank you.
If we could get one last final round of applause. All right, so for our last but certainly not least speaker, we have Dr. Christopher Sowie. Dr. Sowie is a pharmacology and toxicology reviewer at the Food and Drug Administration, and he is an alumnus of the Urema lab here at Johns Hopkins, where he worked on developing different glycoengineering techniques and solutions for treating illnesses and disease. An expert in all things therapeutic, Dr. Sowie knows the ins and outs of the approval of cell and gene-based therapies and is a frequent guest speaker for various classes at Hopkins. Dr. Urema holds him in the highest regard and has only fond things to say about Dr. Sowie, and I've personally very much enjoyed his talks. Ladies and gentlemen, it is my honor to introduce Dr. Christopher Sowie.
Thank you, Michael. You can hear me. Is that correct? Okay.
Yep, we can hear you
Let
fine.
me just share my screen. Okay. Okay. Do you see? Sorry. Do you see the big screen or the? You see the full screen? Is that correct?
We see the presentation like on the PowerPoint mode. Yeah.
Oh, shoot. Why isn't? Sorry. Usually we use the. Okay. How about now?
Oh,
okay.
Yep, that's perfect.
Thank you, Michael, for that kind introduction. And I'm glad to be here, and thank you for the invite. I love talking to students and seeing the research. It's very exciting. So as Michael said, my name's Chris Sowie. I've been with the FDA for about 3 years now. And I currently work as a preclinical reviewer or pharmacology toxicology reviewer in the Office of Tissues and Advanced Therapies in the Center for Biologics Evaluation and Research, or CBER, in the FDA. And today I'd just like to talk to you about preclinical safety assessments for cell and gene therapy products. So this talk is probably going to be a little different than what you might be used to because we will be focusing more on the translational aspects for cell and gene therapy products. And right now you might be wondering, what does cell and gene therapy have to do with biomaterials, but I will try my best to work into the discussion of how biomaterials play an important role for these kinds of products that we often see at the FDA. So here's a little outline of my presentation today. And I've just first like to start with just discussing the organization of the FDA and just talking about, where does my product go when I submit an application to the FDA? Because it's going to be very important if you're thinking about translation just to understand the layout of the FDA and understanding what kind of offices and centers are in the FDA, because each kind of center is going to have some of their own regulations. So it'll be important to understand just the organization of the FDA. And then following that, I will talk about some of the interactions with the FDA and CBER that are available, and how you can interact with us at the FDA to hopefully help guide you towards successful translation of a investigational new product. And then after that, I will discuss some preclinical considerations for assessing the safety of cell-based products, and then following that for gene therapy-based products. So here's a little map of the FDA, and I've just highlighted on this slide some of the three main centers at the FDA that some of you might be familiar with. So the first being at the bottom there, we have the Center for Drugs Evaluation and Research, or CDER. And CDER is going to get all the small molecules, the antibodies, so those kinds of products. And then the Center for Devices and Radiological Health, or CDRH. So many of the products or types of technology that the audience might be interested in would fall under the jurisdiction of CDRH. So if you have some sort of like biomaterials that might serve more as a device, they're going to be regulated by CDRH and might fall under types of regulations such as 510K. I don't know if you've ever heard of that kind of type of regulation, but unfortunately I won't be able to answer questions for how CDRH regulates their products, but I will try my best to at least talk about how CBER regulates our products. So I work in CBER, as I mentioned, so the Center for Biologics Evaluation and Research, which is located in Building 71 right there on the right. But just to give you a sense of how the center is laid out. So we have the center, and we have three main offices in the center. So we have the Office of Blood Research and Review, or OBRR. And then next we have the Office of Tissues and Advanced Therapies, or OTAT, which is where I work. And we have the Office of Vaccines and Office of Vaccines Research and Review, or OVRR. So some of you may be wondering, where do these all these COVID vaccines you read about in the research in the news, where do they go? So they actually do go to us in CBER, in the Office of Vaccines Research and Review. So those are, my colleagues down the hall, OVR. They're very, very busy right now. However, we in OTAT also do get many products for treating COVID. So I've, personally reviewed convalescent plasma, ideas for convalescent plasma, stem cell therapies for treating COVID. However, it's sort of strange. So, sometimes we do encounter nanoparticles that might contain mRNAs that encode for antibodies. So if they contain codes for antibodies against COVID, they will come to us in OTAT. But there's a lot of overlap between OTAT and OVR right now for reviewing all these COVID treatments. And, never in my wildest dreams would I have ever thought, I would go straight from doing my PhD to, on the front lines here of this once in a lifetime pandemic. So it's certainly been very interesting times in the office, and super busy right now. But just to give you an idea of the diversity of CBER OTAT related products, in our office, we get all of the gene therapies. So, all those ideas for CRISPR, microbial vectors, non-viral vector types of ideas, but relevant to the biomaterials audience. So I will assume that nanoparticles might fall under the interest or realm of biomaterials. So if you contain those nanoparticles that are used to deliver mRNAs, they will fall under the jurisdiction of OTAT and will be likely regulated as a gene therapy. We also regulate all of the stem cell products, such as embryonic stem cells, products having to do with iPSCs, and so again, relevant to the biomaterials audience, if you let's say have a biomaterial for encapsulating some type of stem cell, or some type of biomaterial for repairing tissue damage that contains cells. So if the biomaterial contains cells, it will come to us in OTAT and likely to my desk. Some of the other types of, I guess, applications we often see that contain novel biomaterials are often devices or products for xenotransplant, or sorry, devices or things for like wound repair, especially for like burns or like diabetic ulcers. A lot of the the grafts for replacing a blood vessel too that might contain like stem cells or some type of cell will again be regulated by OTAT. And as I mentioned before, like CDRH has their own kind of regulations and CBER has their own regulations. However, we are not shut off from each other. So a lot of the devices that might contain biomaterials, we will have interact back and forth between CDRH, and CDRH might also have questions for CBER. So we're not closed off from each other. It's a very academic kind of environment at the FDA. So we have experts in all sorts of different fields, and it's very collaborative when we review these kinds of products. So this is just to give you an idea of the growth of cellular therapies from the 80s up to 2019. So as you can see, around the turn of the new millennium, we started to see a lot of increase in the number of applications for cell-based products. So they've almost doubled from the early 2000s up to now. Unfortunately, I don't have the numbers for 2020 yet. They're still being crunched, but I can assure you that they're probably at least this high, or at least as high as about 96 to 91, or possibly even higher. And there's also been tremendous growth within the realm of gene therapies. So at least from the mid 2000s up to around to now, I mean, the number of programs that have focused on gene therapy ideas is almost increased by 5-fold. So, if you had to ask me, we're sort of in this golden era right now of cell and gene therapy based products. So it's really starting to take off right now, and we certainly do feel the effects of all this success in cell and gene therapies in the office right now. So moving on, again, I'd just like to talk about some of the earlier interactions that are available with the FDA to help guide to help guide you towards a successful translation of your idea. So many of you might be aware, of course, to market a product, you're going to have to pass phase 1, 2, and 3 clinical trials. And once you complete a successful phase 3 trial, you're going to file for what's known as a biological licensing application, or BLA. And once you obtain a BLA, you are legally allowed to sell a product in the entire country. But that's way down the road. Right now, many of you are probably focusing on preclinical development, or the portion that I've circled here on this slide. And during preclinical developments, the FDA actually has 2 types of interactions that are available, both interact meetings as well as pre-IND meetings. And again, the goal for these two early interactions is to hopefully guide you towards filing a successful investigational new drug application, or IND. And what an IND is it's just basically permission from the FDA that allows you to conduct a study on humans in phase 1 trials. So it's required to obtain an IND to begin phase 1 studies. And when you submit an IND application, the FDA is typically given 30 days to review your materials and make a hold or no hold decision. And during this time, we're really trying to focus on whether or not the safety and rights of study subjects are adequately protected, based on all the information you have submitted. So as I mentioned, one of the first early meetings that are available during the preclinical development stage are known as interact meetings, and this just simply stands for initial targeted engagement for regulatory advice on CBER products. And the goal at this stage is just to get very, very early feedback on your product development. Typically at this stage, you might have done some in vitro work, maybe one animal study or not, and you're just looking for guidance on, what does the FDA expect now that I have this idea in mind. I have very early POC data available, and the purpose of this meeting is just to serve as an informal scientific discussion. So you introduce the FDA to your idea, all the reviewers in CBER to your idea, just to obtain early feedback. Typically, a meeting package for this, a package for this meeting would, it's probably going to be on the order of about 20 to 30 pages. Again, very informal, just to introduce your idea and obtain early feedback. And the timing of this is, again, when you've obtained that early POC, no real safety data yet, but you definitely want to approach the FDA for an interact meeting prior to conducting any pivotal preclinical safety studies. And if you are looking for additional guidance on how to potentially approach the FDA for interact meetings, there's a link right there at the bottom. And moving on from that, sorry, one thing I should mention too about the interact meetings is that they can be very difficult to get. So the reason being is that our resources are very limited in the office, and interact meetings are typically reserved for highly novel products that we've most likely have never seen before. But I will say this, if you have a biomaterial that contains cells, those kinds of products tend to be very complex and very novel. So if you attempt to approach the FDA for an interact meeting with that kind of product, I would hazard to guess your odds of obtaining a interact meeting would be fairly higher than some of the other kinds of products, just because these biomaterials containing cells or tissues are very complex. But moving on, so after a potential interact meeting, you have a pre-IND meeting, which is a non-binding but formal scientific discussion that involves all disciplines of review at the FDA. So you have CMC or chemistry, manufacturer and controls, who are going to discuss how you manufacture your product. And then you're going to have pharmacology or toxicology, or the preclinical reviewers, as well as the team from the clinical team who's going to discuss your clinical development plans. And again, this is formal. So typically at this stage, you've conducted some animal studies, you've finished up, your in vitro POC, and you're contemplating your design of definitive safety studies. And your meeting package at this stage is usually on the order of about 50 to 100 pages, somewhere in there. So it's more formal. You have polished up data that you want to present to the FDA, but you're not submitting full study reports, just summaries of data that you've obtained thus far. And the timing of this pre-IND meeting is basically prior to conducting, as I said, those pivotal safety studies that are going to be very complex and expensive. So moving on, sorry, one thing again that I should mention is that just based on anecdotal discussions at the FDA too, if sponsors who go through the whole process of interact meetings to pre-INDs, they're typically pretty successful at getting a IND greenlit to begin phase 1 testing. I think it's been mentioned before that sponsors who go through this process are on the order of maybe like 70 to 80% successful at getting their IND greenlit to proceed. So it's definitely worth your time to come in for those interact and pre-IND meetings if you're thinking about translation. Moving on to the next part of my talk, we're just going to briefly discuss some preclinical considerations for assessing the safety of cell-based products. Just to give you a sense of how the FDA evaluates preclinical safety for cell and gene therapies, we have tons and tons of guidances for industry if you're not familiar with that. But with relevant to CBER and OTAT, and especially for pharmacology or toxicology or preclinical review, the guidance right here shown on the screen is sort of like the de facto gold standard of what we think about when we're regulating cell and gene-based therapy products. So again, if you're thinking about translation, just search around the FDA's website. There are guidances for many, many different kinds of products. So you should become familiar with these guidances for industry. Again, if you're thinking about translation, because there are many different kinds for all sorts of different products, and hopefully you can find which one's relevant to you, and then read these guidances to figure out, how should I go about designing my preclinical development program. But some initial considerations for preclinical testing should include an understanding of the mechanism of action of my product, of course, and what are some of the intrinsic properties of my product, how is it working, and is this mechanism of action going to be relevant to the clinical population that I'm targeting. So of course that sort of makes sense, but you'd be surprised about what kinds of ideas sponsors come in with. So just spend some time thinking about how's my product working and who am I targeting. And then once you have a good idea for that, you can start considering appropriate animal models for selection to begin preclinical studies. And also, do some digging, and also consider, of course, what kind of data is already out there. Because you don't always have to reinvent the wheel. Just consider, what kinds of ideas have been proposed before for the intended target population. What kinds of data or examples exist for the clinically intended route of administration, and also consider, if other types of similar products have been administered to human subjects before. So just consider, some of the background as well, or maybe there is none. So, again, a lot of these products that contain biomaterials tend to be more complex. So maybe there are no examples to go off of. So just to give you a flavor of some of the potential safety concerns for cell-based products, you should try to characterize how cell properties might change due to ex vivo manipulation. So are you expanding the cells prior to seeding them on a biomaterial scaffold? Is the encapsulation of a iPSC, for example, going to affect the way it differentiates them? These are all things you have to characterize. Is there going to be potential for inflammatory immune response to the cell that you're seeding on your scaffold? And of course, other issues that are, sort of maybe common to some of the more stem cell based products, such as inappropriate cell differentiation, issues with tumorigenicity, or risk of unintended distribution of cells to non-target sites or tissues. And one thing to not overlook as well are risks of the delivery procedure itself. Is some sort of surgery required to implant the scaffold, or are you injecting some kind of biomaterial to a certain location in a person? So you want to mimic that as best as possible in a preclinical study. Just some considerations for preclinical testing of cell-based products. So again, your POC should, of course, demonstrate evidence of therapeutic effect in animal models. So hopefully, there's going to be animal models available either through genetic manipulation, or maybe you can induce some sort of insults to sort of recapitulate the injury or disease, and hopefully those types of models can be used to establish proof of concept. So as mentioned before, the fate of the cells that you might be, seeding onto a biomaterial scaffold are going to be important. So how long do the cells persist? Does their phenotype change over time? Do they proliferate? And again, are they affected by that route of administration? So hopefully you can model that in those preclinical studies as well. So ultimately, as I mentioned, at the pre-IND stage, you would come in prior to conducting a definitive safety study. And this slide right here is just describing what a typical definitive safety study would encompass. So they're going to contain standard toxicology endpoints such as mortality, clinical observations for change in behavior, body weight or appetite of the animals, as well as clinical pathology parameters that are specifically relevant to your product. So a typical safety study is going to include studies for changes in hematology, coagulation, serum and chemistry, urinalysis, et cetera, et cetera. And then what are quite important, especially for novel biomaterials, are going to be terminal assessments. So macroscopic exams upon sacrifice, measuring organ weights, and especially histopathology. Typically during a definitive safety study, you're going to have a well-qualified veterinary pathologist, hopefully a qualified veterinary pathologist evaluating the histopathology following implantation or administration of your product. Design parameters that we also think about during those pre-IND meetings are, when you propose your safety study, we're looking at, does the study contain an adequate number of animals per group for study robustness? We just want to make sure, you're not conducting a study that contains not enough sample sizes to draw any type of meaningful conclusions, because at that point you'll be wasting animals. At the same time, we don't want you using too many animals, because we are very sensitive to the 3 R's for reducing the number of animals. And the definitive safety study might also have to consider multiple sacrifice time points, as well as propose a sufficient study duration to evaluate both acute and long-term safety endpoints. So if you can sort of see, this is getting very complex. You have, probably controls, multiple treatment levels, and then you're going to have multiple sacrifice time points, different types of duration, and then at each one of those time points, you're going to have all these standard toxicology endpoints. So that's why you definitely want to come to us during those pre-IND meetings to discuss design of your definitive safety studies. And these studies tend to be the most expensive and complex ones at the preclinical stage to conduct. So some additional comments with respect to the selection of animal species. So CBERs, again, as I mentioned before, it's very important important to understand where your product is going, because CBER is a lot different than, say, CDER, so the Center for Drugs. CDER is a lot more, or sorry, CBER is a lot more flexible in that there's no default to the use of non-human primates, and there's no default to requiring two animal species, two different species of animals. So that's somewhat, actually a lot different than CDER. So again, it's very important to understand how each center regulates their products. And when you're considering animal models, it's also important to understand the limitations of the species or the model used. So, if you're trying to inject a certain volume into an animal, or sorry, you're considering like injecting a volume into a human, but so you can't do that in a rat just because it's so much smaller. So you just understanding the limitations of each model, each species or animal model used is important when you're designing your safety study. And you should also provide scientific justification for the type of animal species or model that's used. So, if you propose to use a non-human primate, you better have a very good reason for using those types of animals. So, just justify everything that you're doing when you're proposing your definitive pre-safety, or sorry, your definitive safety study. So moving on, so that was preclinical considerations for assessing the safety of cell-based products. But next we will just briefly discuss gene therapy products. So there's a lot on the slide here. There is a lot of overlap between cell therapies and gene therapies, just in terms of thinking about safety design and animal selection. But relevant to gene therapies, you do want to think about biodistribution and persistence. So I know right here on the slide I put vector biodistribution and persistence, but if you have a nanoparticle product that contains a gene therapy, I can tell you right now that biodistribution and persistence of the nanoparticle itself are going to be important safety considerations during review. Also important is the potential for germline transmission or developmental and reproductive toxicology, or DART, especially if you have, say, like a nanoparticle that contains mRNA encoding for a CRISPR-based gene editing product. So potential for germline transmission of that type of product will be very important during your safety studies. Depending on your product, a long-term follow-up will be required. So gene therapies typically require 15 years of follow-up, and this this will probably fall under the realm more of your clinical development program, but you do have to consider, when you're administering a gene therapy, that significant follow-up will be required out to 15 years. And legally, if you say have a company and it goes out of business or something, that does not absolve you of following up with these people who you've administered this gene therapy to. So long-term follow-up should definitely be considered in your product program development. Sorry, the last point is probably not relevant, but shedding concerns. If you potentially had a viral gene therapy, there would be issues as well related to shedding. But for biomaterials, that might be less relevant. So with respect to animal selection, some of the additional things you should think about, is my animal model or species permissive to the infection or viral replication? But again, not in the case of biomaterials. However, I would say, maybe if your biomaterial contain, like, again, a gene editing type of approach, your animal model or species should be amenable to that gene editing compared to a human. the tropism of, let's say, a nanoparticle or immune response to a nanoparticle in a animal should hopefully be representative of what will occur in a human. So, for example, many people know lipid nano or something like a lipid nanoparticle might contain like pegylated lipids, for example, and it's well known that pegylated lipids induce anti-drug antibody response. So hopefully in humans, I mean, so hopefully an animal model as well will sort of induce that kind of same immune response, just so you can get an idea of, um, the rapidity and onset of the development of ADAs against a biomaterial. Again, similar to cell-based products, scientific justification should be provided for every animal model selected for a gene therapy. And I won't go over this slide here, but there's additional concerns, of course, for insertional immunogenesis or chromosomal damage, especially if you have like a gene editing product. This is just a framework of how you go about evaluating risks for gene therapy products. So if this is of interest to you, you can look at the link below for more guidance on how to evaluate the risks of gene therapy related products and the potential for delayed adverse events. And finally, I would just like to talk about, what do you do with all of this preclinical data and testing? Ideally, it's used to inform the design of a clinical plan. So if everything goes according to plan, hopefully you can identify a first in human dose that's extrapolated from the safety data from your animal studies. So, it might be a little bit strange to talk about dose if like you have a biomaterial scaffold with cells. However, this it would hopefully translate to maybe you base it based on like surface area or size or something like that, versus let's say a nanoparticle, which might be based on, let's say, a mg per kg type of dose. But anyway, the preclinical studies are used to justify that first in human dose level. And preclinical studies are hopefully used to also inform on dosing regimen. So how often am I administering a product? Is it once, twice, or multiple, even more times that? So ideally, when you're conducting your preclinical studies, you will use a regimen that mirrors exactly what you tend to do in human subjects. You also want to establish feasibility and reasonable safety for that product administration procedure. So that's one area that sponsors may often overlook, is just the risks for the delivery procedure itself. So that's something you definitely want to consider while you're testing these kinds of products. And additionally, some other ways preclinical development can inform the proposed clinical plan, or to support patient exclusion criteria, as well as identify potential toxicities and physiological parameters that help guide clinical monitoring. So with that, I will close. I think I'm out of time anyway, but here is our contact information for both myself as well as for OTET. With that, I'd just like to close and say thank you. And if there are any questions, I would be happy to answer.
Thank you again, Dr. Sally, for presenting on your work at the FDA, especially in terms of, or especially, I guess, in light of the recent COVID vaccines and how they're involved with your work.
Thank you. Yep.
Does anyone have any final questions for Dr. Sally? If not, I think let's give him a final round of applause to thank him for being present with us and helping us out and informing us of all these important things.
Thank you.
All right, so that concludes all of our guest speakers for the Mid-Atlantic Biomaterials Day. And as this overall conference winds down, we have just a few last final things to take care of. Specifically, we'll start off with the presentation of awards. This year's awards were generously funded by the Alumni Association for the Best in Show and runner-up rapid fire talks and poster presentations. First, starting off with the runner-up, we would like to congratulate Ikshu Pandi for her research poster presentation.
Thank you.
And then finally, for our Best in Show award, we would like to congratulate our visitor from Arizona State University, Subhadeep Duda. And we will all be in contact with you to get the distribution of the awards nailed down as well. And now for our closing remarks. I would once again like to thank everyone that was able to attend this event and experience the world of biomaterials with us today. Every year, we collaborate with other universities and Society for Biomaterials chapter in hosting this event. And every year, this event keeps getting better and better and more successful. This year, especially, I'm honored to have had the opportunity to still host this conference, despite everything that's happened in the last 12 months. And I'm glad to see that everyone was able to join us today, with special thanks to the other SFB chapters from universities across the country. Something that you might have also noticed this year is that many of our speakers are JHU alumni. This year, we wanted to emphasize the connections abroad and at home that connect us through the biomaterials world, no matter where we are. And it's truly been inspiring for me to hear both about their talks and about their journeys that led them to where they are. As someone that's soon to be an alumnus himself, I hope to similarly inspire future students, just as all of the alumni that I look up to have taught to and motivated me. In the planning of this conference, we would especially like to extend our thanks to the crucial individuals and organizations that help us successfully host our event. First, we would like to thank our sponsors, the Alumni Association and the Department of Materials Science and Engineering, who generously provided us with the resources necessary to both host this conference, as this event would not have been possible without their support. We would especially like to thank the administrative manager for the Department of Material Science, Ellen Lee Bao, and the Departments of Biomedical Engineering, Biophysics, Neuroscience, Molecular and Cellular Biology, and many others that helped us advertise this event to the undergraduate population at Hopkins. Additionally, our advisor, Dr. Hai-Tran Mao, greatly helped us with the planning of this event, and he has always been an amazing advisor, both for the club and in life. Finally, I would like to personally thank my executive board members for helping with both the planning and execution of this event. They've truly risen above and beyond any station that I could have possibly given them, and they have been working tirelessly to make sure that everything has been in the right place, and this event definitely would not have happened without them. Here, I am merely the face of the SFB chapter, but in a sense, I really am just a face. Everything that was involved with executing this event all came down to the board's dedication and commitment to making this conference happen every year. Although I am undoubtedly sad to see myself leave Hopkins, I know that I'm leaving our SFB chapter in good and capable hands that can only further heighten and enhance our club for the future. To all the attendees, again, thank you. We want to thank you so much for attending Mid-Atlantic Biomaterials Day this year, and we hope that you will come again in the years that follow, whether they be in virtual or in person.